and we'll alternate those. And we'll alternate those with internal speakers. But at the moment, I'm happy that I've got a really great roster of external speakers coming for the 2022-23 series, and it's wonderful to kick it off with Rafa. So as I mentioned, so Rafa is going to give us an amazing talk today on untargeted metabolomics, mapping the cancer metabolome to identify therapeutic vulnerabilities. And Rafa actually earned his PhD in chemistry at Vanderbilt University. And I have been known to say my favorite people are chemists. There's many chemists in the room who will attest to that. He did the PhD under the guidance of Dr. John McLean and focused on the analysis of complex biological samples using ion mobility mass spectrometry, coupled to advanced pre-ionization separation techniques, such as supercritical fluid chromatography. And then he went on to a postdoctoral fellowship at the Scripps Research Institute in La Jolla under the tutelage of Dr. Gary Siustak. And there he focused on advancing the analyses of highly complex multivariate data in untargeted metabolomics, as well as solving challenges in the identification of unknown metabolites and their corresponding biological functions and activity in disease, such as cancer. So Rafa joined the Donnelly Center with a beautiful suite of Orbitrap, mass spec, all kinds of cool equipment, which I'm very impressed by, as an assistant professor in the fall of 2020. So trainees, this is someone who's quite close to all of you. And so I hope that you took advantage of chatting with Rafa about career building in science today. And really what he's focused on at the Donnelly Center is the application of mass spec and bioinformatics technologies to characterize the metabolome and to discover novel endogenous metabolites that are associated with specific phenotypes. And Rafa and I in particular have been talking a lot about an approach that we took when we set up the original SCCRI and now the CDCR as a comparative biology approach and looking at the difference between normal and cancer, which is often very instructive. And I think Rafa may be doing some very exciting work in that regard with metabolomics as well. So please help me give a warm welcome to Dr. Rafa Montenegro Burke, and let's hear from him now. So thank you, Rafa.
Oh, thank you. This is the best introduction I've ever had. Thank you. Well, thank you for the invitation. I'm honored to be, giving a talk here, and also being the first person to give a talk. It's been really great coming here, meeting some, I don't want to say old faces because I've seen many of you in Zoom, but it's been great to see you now in 3D and also some new faces. It's been really, really fascinating, and like some of the discussions made me think about it, like some of the projects that we're working on, like really think about in very different ways. So yeah, it's been really great. So as Sheila was saying, I'm a chemist, and my lab focuses on metabolomics using mass spec. We develop resources and methodology to better profile and understand the metabolome and metabolism, and we then collaborate with many biology labs, and for today's talk, I'm going to talk about more some of the collaborations that we've been working in Sheila's lab and doing a lot of work with Will. But I'm going to start just to talk with a project that we published, like the last project I worked on my postdoc, just to show you what metabolomics is and how we go about looking at data. And then in the second part is this project that we're working on that is different in terms of like how we actually approach and how we actually do metabolomics. So everybody can hear me in the back is fine? Perfect. So first, let's just start with what is metabolomics. As you're probably aware, genomics, is a discipline where we analyze and profile the entire set of entire DNA of an organism or of a cell. Metabolomics is the discipline where the complete set of metabolites are analyzed in a cell or an organism. Now, what is metabolism? It can be a little bit of different definitions depending who you ask, but roughly it's small molecules that are not DNA or proteins or RNA that are below 1,500 Daltons. Sometimes it can be bigger, and essentially are molecules that are used by organisms and are metabolized by them. Why are we interested in metabolomics? So the main advantage of why we're interested in metabolomics is that it is at the bottom of the omics cascade, which you can see here. Usually is, genes become proteins and then proteins metabolized metabolites, right? So being at the bottom of the omics cascade is really close to the phenotype, so it can give us a really good idea of what is happening now. If you think genes and the time it takes from genes to become proteins and so on, there's a disconnect between them. So genes can tell you what could happen. Transcriptomics, proteomics is like what is going to happen, and metabolomics can tell you what is happening at this particular moment. Oftentimes we think that metabolites or metabolomics is exactly just at the bottom and is the end result, but we also know that small molecules metabolites can operate, can regulate biological processes upstream, right? If we think genes are made of like nucleotides, proteins are made of amino acids and so on, there are other regulatory processes as well. One thing that's really interesting, or like why metabolomics can be really, really fascinating, is that like a small modification in a gene, as you can see here, can result in the change or the dysregulation of a broad range of molecules, right? When we think about just a gene, then to protein is a very linear path, but once you get into metabolome, there's metabolic rewiring, and we see a broader change of the entire metabolome. So it's a little bit trickier to decipher those things. So when we do metabolomics being the newest of the omics disciplines, it's always going to be compared to genomics and proteomics, right? So when we look at the numbers, in genomics, the human genome has roughly around 20,000 genes, and they're composed by 4 nucleotide bases, right? When we look at proteins, again, 20,000 proteins, now we're starting thinking about proteoforms. They can be up to 1 million, depending on how you count, but they're made out of the exact same 20 amino acids. When we come to metabolites, we really have no idea what the number is. It's very, very complex. We don't have a map for it. So that's one of the challenges or more difficult parts in metabolomics. We don't know how many are there. And even in the human body, we eat different organisms, right? Microbiome is really, really vast and complex. So there's a really broad chemical diversity, and that's one of the main challenges in metabolomics. I'm going to talk a little bit about more in depth about that in the next slides. So again, my team is a multidisciplinary team with different expertise, where we use analytical chemistry, bioinformatics, a little bit of chemistry and synthesis to really probe biology. And the first project I want to tell you about involves IBD, but first I want to go a little bit more in depth into like what metabolomics is, or the techniques that we use. We rely a lot of liquid chromatography and mass spectrometry, and there are 2 main approaches. The first one is targeted metabolomics. In targeted metabolomics, you actually design and set up your instrument and your assay to profile and quantify a handful of metabolites, between a dozen, 2 dozens, no more than that. And it gives you really, really good quantitative data. The disadvantage is that you miss out on a lot of other metabolites. And that's where we, with Untargeted metabolomics, using different chromatography techniques and different instrumentation, we can detect tens of thousands of different metabolites. So it gives us a really good picture of what the phenotype looks like. So again, I want to tell you about this project because it covers different aspects of why I'm really interested in metabolomics. Inflammatory bowel disease, as you probably know, it's an umbrella term for many diseases, but it can be generalized, described as inflammation in the gastrointestinal tract. So there's a really high number of people, both in Canada, US, around 2 million that suffer from IBD. And it doesn't affect only adults. Around like 90,000 of those patients are below the age of 18, really. And the trend is going to increase given different factors, but it's expected around 8 years. When I first put this slide together a couple of years ago, it was in 10 years, now it's 8, so we're closing in fast. Around 1% of the population, particularly in Western society, is gonna have some type of IBD. And the exact cause is not known. There's been several links to genetics, exposure, and so on. And we know that around 1 in 5 patients has a relative that also has IBD, so there's a genetic component to that. But one of the things that is really interesting is the diet and the exposure. The exact a cure, we don't have a cure per se for IBD, but different treatments involve anti-inflammatory drugs and so on. But one that really caught our attention a couple of years ago was that elemental diets, which are composed by very simple amino acids, sugars, and fats. You can think of a protein shake that you just did with like opening a bunch of jars from Sigma Aldrich and just adding all the different components and mixing it. Really lowers relieves some of the symptoms and does really help with IBD. So if you think about it, all those molecules that I told you about are metabolites. So there's a clear link. The diet, change of the diet to something very simple that actually regulates or down-regulates the inflammation in IBD. So we thought this is a really perfect case, a really perfect example where we can use our technology, where we can profile tens of thousands of molecules in one experiment to try to understand what metabolites could be regulating or affecting inflammation. So this is the general experimental design, and I'll just go briefly over it. We have our samples where we look at the feces, the cecal, and the colon content on this IBD model of wild type versus a RAC knockout with a T-cell transfer. And we look at the feces and cecal and colon content, not the most fun type of sample collection, but it was really informative. We did some sample preparation, and we rely a lot of LC-MS mass spec to detect all these molecules. Now, after that, there's a good amount of data processing using very specialized bioinformatic tools, and then we get something that's called the metabolome output. So at this stage, we don't know what all these metabolites are. All we have is, if you ever done some type of omics experiment, you get this huge Excel spreadsheet, and then you gotta go and figure out what all that stuff looks like. You do PCAs and all these things to try to make sense out of that, right? But we don't know what each of all these, we call them features, is again, a mass with a retention time. We don't know what they are. So we spend a good amount of time here looking at the data and prioritizing what we want to identify. We spend a lot of time looking what we want to go back and fragment, and those are exactly the molecules that change. If something didn't change, again, we're not that interested in that. And that is the general workflow in metabolomics. Makes sense. To identify them, we then go back, we analyze the sample, and we fragment molecules. So again, the features that we're interested in. And here I'm showing you how they fragment, right? They fragment in a very specific way. You can think of those like as a fingerprint. Each molecule has a very defined fingerprint, and we rely on databases so that we can do these comparisons to be able to identify these molecules. Again, our databases are not complete since we don't even know how many metabolites are there. So that's a limitation. And this part is actually the bottleneck in metabolomics. We can detect many, many molecules. We don't know what they are, and the identification is challenging. But once we do that, we get a couple of candidates, and we can call these biomarkers. And if you look at metabolomics, most of the application is for biomarker discovery, which is great, but it doesn't really tell you what the function of these metabolites are. Are they just casual, or do we actually know that these metabolites are inducing a change in the phenotype? So that's an area that we're particularly interested in. So I want to show you just a list of the potential biomarkers, and I know it's a very busy slide. I don't expect you to look at all the different molecules, but the take-home message that I want to show you is that with this technology, you can detect a broad range of molecules and changes and identify them. We can see some fats around here, some peptides, vitamins, forgot what the name is, bile acids, amino acids, some other energy-related molecules. So it's a really broad range of molecules that change. In yellow would be molecules that drop in the IBD cohort, and in purple are the ones that are upregulated. So it's a really broad change, and there's no really distinctive pattern, right? And there's many, many other molecules that change that we're not able to identify. So the approach, what we were interested more, or not necessarily just getting these biomarkers, but then actually, what are they doing, right? So we move on and try to screen these metabolites to see if they're actually playing a role in inflammation. Essentially, the question we're asking is, can some of these metabolites tip this balance that I'm showing you here, tipping it towards senescence or tipping it towards inflammation? So the first experiments that we did for this were using cell culture assays. And I know also a very busy slide from the paper, but we use primary macrophages and THP1s. And essentially what we're asking is, if we induce inflammation, can some of these metabolites reduce the inflammation or actually would enhance it? And we tested many of them with several of these assays. And what we noticed was there's this metabolite right here, phytosphingosine, that dampens inflammation, very close to a level of the bar above dexamethasone, which you know is a very strong anti-inflammatory drug. So this was really interesting. This is not only effective in an IBD model, because these are like cell cultures with very normal, cell culture conditions, just macrophages. And here I'm showing you the structure of phytosynthesis. And I'm saying with Ali, I wish I would have talked to you before I prepared the talk, because I would have talked more about this and what we're doing with this next. I'm happy to chat later on, but yeah, this is the molecule that I was talking to you before, phytosynthesis. And when we look at that, again, it was a molecule that was downregulated in IBD. So now we have a molecule, a metabolite, that is at lower levels in IBD, but it's also anti-inflammatory. So, this could actually be inducing, not alone, but it could actually be contributing to the inflammatory state in IBD, right? So this gave us confidence to go back and test it in vivo, and that we use an IBD model as well, and to look at how this metabolite can regulate inflammation and also membrane permeability, which is one of the major challenges in IBD. The membranes become permeable, you can absorb less nutrients, and it results in body weight loss. So one of the markers we look at this in vivo model was MCP1, monocyte chemoattractant protein 1 that recruits monocytes. And what we found here was, here in white, in the white bar is the vehicle. In the IBD model, we see an increase. This was looking at the mRNA levels in the colon tissue. And when we treat with phytosynthesis, we lower the levels almost pretty close to the levels of the control. Now, again, this is in vivo. Also, the membrane permeability, two markers that you can use for that is occludin and tight junction protein 1. These two proteins keep the membranes tight and control the permeability. And similar to the MCP1, what we observe is a recovery, not as high as inflammation, but it does improve, shown here, the levels for both occludin and tight junction protein 1. So taking all this together, we know that the phytosynthesis not only is anti-inflammatory, but it also helps protect the colon from inflammation. It actually can tip the balance towards senescence. Now, I was telling you that in target metabolomics, we can profile tens of thousands of different molecules, and we generate those long spreadsheets that you probably are very familiar from other omics disciplines. But how many are known, or how many do we actually know, really? It's a really hard question, and that's us actually, this field has gained momentum, this field has gained momentum in the last couple of years, and it's called the dark metabolome. So these are molecules that we can detect, but we don't know what they are. And it comes from the term from dark matter from astrophysics. I'm sure most of you have heard about that. And to get an idea for you, if we did get a drop of blood from all of you, at least 50%, if not less, we will not be able to tell you what those molecules are. And that is the main limitation in metabolomics. But it's a really fascinating field, because if you think about it, if we can find some metabolites that are dysregulating or are affecting the phenotype, when we go and discover these new molecules, we're going to gain so much more information, right? That we're really pushing the boundaries to really identifying completely new biological processes. So depending on who you ask, you'll get different results or different numbers in terms of how much we can identify, but around 2 to 10% in metabolomic experiments is what we can actually identify. So this is where the work that we've been working on more recently in the lab, and we're starting to think about this in a more philosophical way. How much do we know about the human metabolome? If you think about like what I showed you before with the numbers, yeah, we have a number of genes, we have all this information, right? When it comes to metabolites, we know a couple of structures, we know a little bit of pathways, but do we have some functional information? The answer is not really. So we're starting to think about it this way. If we think about the databases that we have for genes and proteins, we have databases that are more like encyclopedia, that are more descriptive, like UniProt, FAM, and so on and so forth, where you have some information about this particular protein and gene and what it does. And we have the same thing for metabolites, like Mon HMDB and databases where we have the list of all these metabolites. Now, another type of database could be thought more about the atlas or that contains more quantitative and functional information. And you probably are familiar with Human Protein Atlas, DepMap, for sure, CellMap, and so on, right? Which gives you really, really cool information about where things are localized or how important is this gene. But for metabolites, we will have nothing like that. So we thought, okay, we should go for it, and we took a Human Protein Atlas as a good model to look and to generate this type of resource for the community. And we started the Human Cell Metabolome Atlas in the lab at the beginning of this year, and we've made some good progress here. And I'm going to show you what we've done so far generating this atlas and how there are different examples or different projects that we have going on with this particular resource. But I'm going to focus more on one that involves some of the collaboration with Sheila and Will. So the approach we took was, okay, can we profile as many cells from different organs? And we are culturing these cell lines in very specific conditions with the same media or FPS whenever possible, same passage, and so on and so forth, and collecting it as close as possible. Using internal standards and so on and so on. We do LC-MS mass spec, but now the approach that we took is different. Before I show you, we get the metabolome output, we do data analysis. What we're doing now is try to identify as many molecules as we can. We then collect this into the atlas, but then that allows us then later on to do some data analysis and develop some tools for that. And at the moment, we have around 940 identified metabolites with standards. And yes, we have around tens of thousands of different metabolic features that we still don't know what they are, but it's really fascinating to have this and to correlate it to a particular type of cell. We have different cells that we have here involving different tissues, and we're focusing at the moment more on brain. We generated this resource as a web page that you can go and click and look at the different metabolites in the different cell lines. Like I'm showing you here, for example, this is a ceramide that's mostly upregulating this K562 cell. And you see there's a very specific regulation of this metabolite in these different cells. At the moment, we have these 20 cells, and we're growing. Our hope is in like 6 months to complete what a Human Protein Atlas has, which is 60. But again, this is where I'm very happy to talk to other groups and other researchers if they have other cell lines that would be interested to get metabolic profile and to add to the atlas. And I hope that I can convince you in the next slides why that would be really fascinating. And the more cells we have, obviously, the richer and the data and the better the analysis and the information that we can gain. So again, with this type of resource, we can do many, many different things, such as identify metabolic vulnerabilities in disease, which is one of the things I'm going to talk about later. We can also determine what the function of the metabolite can be, try to understand what the biosynthesis and how it gets regulated, also identify regulatory genes and link them to unknown metabolites, and also link unknown metabolites to a particular gene, and so on and so on. But again, given the audience and the main interest here of the audience, I'm going to focus on identifying metabolic vulnerabilities. And for this, again, working with Sheila. Can I get rid of this? All right, sorry, Sheila. That was not on purpose. That they know what you did. Working with the SIG group and mostly Will, but it's been great in the last couple of weeks. We're going to have to add more pictures here from the same group, looking at other type of science. So please send me your pictures. I can put them in the slide. And we still focus on group 3 medulloblastoma. And as you probably are aware, the proposed cell of origin is neural stem cells. So we first started by thinking in a more simplistic way, in a more binary type of approach. Essentially, are there specific metabolic changes that are unique or like necessary, essential for the medulloblastoma? but not for NSE. But as we continue and develop this resource, a human cell metabolome atlas, we can then ask other type of questions, like, are there metabolic changes that are essential for melanoblastoma, not for NSE, or any of the other cells in the body? As you can imagine, if you find something that is really specific and lets you target melanoblastoma, not NSEs, but then actually all the other cells are affected by that, it's not very useful, right? So it's one of those things that the bigger our atlas is, the better information we can get. So we've found several really interesting trends comparing, particularly, we're focusing more on these NSE 197 and SUMB 002 cell lines, but we see some very common trends with the other NSEs and metabolostomas that we observe. But I want to talk about these molecules, cholesterol esters. They're kind of boring molecules, but it's because we don't know much about them. And it's a really good example to show you how we can think of these molecules not being that important or like not that well understood or characterized. But I hope at the end of the talk, you'll change your mind about cholesterol esters. So here you can see the main structure of cholesterol, and we use very simple nomenclature to describe or to give them names to these molecules. You might be aware with this, familiar with this type of nomenclature, but this is arachidonic acid, and it has 20 carbons and 4 double bonds. So we call it cholesterol ester and 24. So we're going to call lipids based on this nomenclature. So when we first look at this analysis, comparing the NSE and the melanoblastoma, here I'm showing you on the x-axis the different cholesterol esters species. On the y-axis is just their relative abundance, where everything's normalized to NSEs. And we observe really low levels of cholesterol esters in melanoblastoma. Then we noticed there are other unknown metabolic features that were really, really high in melanoblastoma and not NSEs. And what we discover is that these molecules were actually also cholesterol esters, but they have these extremely long, ultra long chain fatty acids, which are so long, they just decided to add an ultra to that. But these molecules are so strange, these lipid species, they're not detected, or not a lot is known about them. And in the lab, some people call them like not the ultra long chain fatty acids, but the super duper long chain fatty acids. And this molecule is not found in any database. If you go to PubChem, a human metabolome database, and so on and so forth, they're not there. So the literature that we're able to find was dated back to the 1970s, I think, and it was observed only in the gland of eyes and what was the other one? Placenta from horses. So not a lot that you can go from for when you're looking at brain tumors. And we were able to identify it because of our approach of Untargeted metabolomics and looking at the patterns and then manually going and identifying what these compounds are. Now, since then we're developed tools to be able to look at this data in a more meaningful way or in a faster way. When we compare different cell lines, we could have different levels of cholesterol esters, but and normalizing can be tricky. But some of the tools that we've developed are how can we compare very, very different cell lines, could be like apples and oranges, but still gain useful information. And what we did is we started looking at this more in terms of the composition, or a way to think about this is if you add up all the concentration for all the lipid species in that class and normalize that to 1, what is the percentage composition for each of these species? And what I'm showing you here on the x-axis is the carbon length chain, so how long the fatty acid is for this cholesterol ester. And on the y-axis is the saturation level, so the number of double bonds. And on the sides, you can see the histogram for the particular lengths and double bonds. So when I'm showing you this, it becomes quite clear that there's a big change in the length of the carbon and a little bit on the saturation, but mostly the length is quite clear. Here you can see it's most of them are found here. Well, here, these are the more abundant ones. And again, these are very unique molecules. Everybody has heard about cholesterol esters, but again, why are they being upregulated in this type of cancer, right? So what the atlas allows us to do then is, can we get more information about how specific these molecules are? And what you can do is you can look at this species and then you can compare with all the other cell lines, and you can see it's very specific to this particular cell cancer. So if I show you this, you think, wow, it's kind of like a biomarker for that particular type of cancer. The fold change is I don't know how high, is huge, almost not detectable in all the other species, right? But another way to look at the data involves how can we do this analysis faster, where we cannot just look at one species at a time, but can we look at it like more broadly? Oh, sorry. So with the help of bioinformatics in the lab, we start looking at this type of generating this type of plots using the Janssen-Chan divergence index. A way of thinking about this is the higher the index and darker the color, the more different it is that lipid class composition to the other cells. So I'm showing you here for the medulloblastoma, and here on the y-axis, you have all the different cell lines that we have in the atlas. At the bottom, you have the different species, the different lipid species. So you have triglycerides here, carnitines, cardiolipins, diglycerides, and so on and so forth. So remember, the darker the color, it just means it's very, very different to that other cell line. So if we look here, cholesterol esters are very dark purple reddish line, very specific for cholesterol. In all of the other cell lines that we have, we find other differences, but this tells us that the composition of these cholesterol esters is very, very different and specific to this type of cancer. So we can generate these plots and immediately calculate these values, and it tells us how different the composition for the lipid species are. So this is all great. You're thinking more right now, well, why should I care about this? Like, why are these things important? We do cancer research here, so please get to the point. And we're starting to think about this in terms of like, okay, if this is something so unique, it must be important. Why would a cell go to the trouble to make these fatty acids that are twice the length, very insoluble, hard to manage? Why? Is there a reason? It's gotta be important. So could it be a metabolic vulnerability? So it has two parts. It has the fatty acid and the cholesterol. The simpler way was first, okay, our cholesterol synthesis is really important for this type of a cell line. And this is not my work, this is Will's work. But what we thought about was, okay, we can target cholesterol synthesis, which is the way, as you know, many people with heart problems take statins. What we do is we inhibit it steps early on in the steps of cholesterol, and you lower the levels of cholesterol in general. And in this viability plot, in blue, you see medulloblastoma, and red, stem cells. It is a little bit more susceptible. It's a little bit of a vulnerability, but it's not really a big window here, right? So this told us that cholesterol is not the real key here, but it was more the fatty acid. But if you think about it, like fatty acids, like how can that be a vulnerability, or like why are they being made so long? So what the atlas allows us to do is go back and look at all the lipid species, in particular triglycerides, which are known to store all the fatty acids in the cells, right? Whatever you need in terms of fatty acids likely is coming from triglycerides, or if the cell is making it, gets stored as triglyceride. So we start to look at the triglycerides as well and compare these two cell lines to understand what is happening to that fatty acid pool. And again, here the nomenclature, the number of carbons, we add them up. So 16, 18, and 16, it's 50, and 0, 1, and 2 equals 3. So this is the nomenclature we use for the carbons. The number for triglycerides, the number of carbons in all the fatty acids is 50, and the number of double bonds in all the chains is 3, right? So we're starting to look at this, comparing again NSC and medulloblastoma, similar to what I showed you, the cholesterol esters before. And I know the change is not that drastic, and in this case, it's not so much for the carbon chain. It's very, very similar. But what we can definitely observe is that there's a change in the saturation. The NSCs tend to have more double bonds than the medulloblastoma, completely opposite to what we observed with cholesterol esters, where it was slightly more double bonds in the cholesterol esters. So we plotted this in a different way to try to like visualize this better, to get a better understanding of what could be happening metabolically. And what I'm showing you here, similarly to what I showed you with the cholesterol esters, the triglycerides, the different species here, where the more saturated ones are here on the left side and more unsaturated on the right side. Medulloblastomas in blue, they're more saturated, and NSC has more unsaturated species. So why is it completely opposite of what we observed in cholesterol esters? we're starting to think about this in more how those long chain fatty acids are actually synthesized, biosynthesized from the beginning. For example, stearic acid, a C18 zero, so with no double bonds, needs to be first, a double bond needs to be introduced there before it can be elongated by these elongases and to make this ultra long chain fatty acid. So our hypothesis was, okay, it seems that as soon as a double bond gets introduced in some of these fatty acids, they're just being elongated. And that's why they're so much lower in the metaloblastomas. So if we stop this particular process here of introducing the double bond, that could be the metabolic vulnerability. Also, there are no drugs for these elongases yet, but it's something that would be really interesting to explore further. But what we try is using a known FDA-approved chemical inhibitor for SCD. And what we observed was that, yes, metabolomics is very susceptible to stopping this process right here. And NSCs has a very, very good window to tolerate that. So when we look at all this, we can think of these molecules as, yeah, we don't know much about them, like boring cholesterol esters, but it can tell you how particular cells are using in some sort of way these metabolites to escape or to maintain cell viability. And it's very specific to each particular cell. And we find always very specific trends in all the samples that we're processing. And some of the other, comparing also group 3 metablastoma, we found other vulnerabilities, some including nucleotides, and we have a paper in revision right now, some of amino acids, ceramides, which I would love to talk more with Ali about that, and inflammation, and glycosphingolipids as well. So by being able to profile and catalog and generate an atlas where we get a better understanding what all these different type of cells from different tissues and cancer, we can really go and understand and develop alternative therapeutics for these diseases. So in summary, I hope I was able to convince you that metabolomics is really a good readout of the phenotype, and it can actually, metabolites play active roles. They're not just like the downstream or the effect of all these genomics and proteomics processes. Also, systemic and comprehensive metabolic profiling and generating these maps help us really discover new metabolic vulnerabilities in disease. And this is not only applicable for group 3 metabolostomas, we've seen others with glioblastomas as well. So it would be great to look into other types of cancer as well. So I think this is the most important slide, the acknowledgements, all the people that have been working really, really hard in the lab. Mohammed has been doing a lot of the bioinformatics aspect of generating those plots, like how we can determine if the composition of a lipid class is different between all the other ones. Brandon's a bioinformatics person that helps us identify molecules. Sunji and Andy identifying and prepping a lot of these samples. Nick, he's a former McMaster undergrad student that I managed to poach from here. Really happy. He was in the chemistry department, and he did a lot of research with Philip Bridge McKibben doing metabolomics. So he already came really well trained. He's doing fantastic work trying to get to single cell metabolomics. And I wish I could take credit for him doing so great work faster, but I think it was all training that he got here. Jihong and Jeremy have been working on identifying and growing a lot of the cells. There are many lipid classes, many molecules. So we've been tackling in teams with Kelly, Alex, and Mathula, which are 3 undergrads in the lab, of generating these maps of like sphingolipids, phosphatidylcholanes, and so on and so forth. So everybody has been working hard on this atlas. I wish we would have more time to show you more of the data and some of the other interesting trends that we observe. But yeah, this is our main project. We're really interested in continuing populating this, and we're looking for other people that are interested in getting their cells profiled. Funding, and thank you very much for your attention. Happy to take any questions.
So guys, help me thank Rafa. This is a really fascinating talk, Rafa, and just so interesting, and so much sort of novel science you've shared with us today. So I want to open the floor to questions. Does anyone have any opening questions? Neil.
Great talk, Rafa. I think there's more than enough people in this room who want to keep you busy. So I was just wondering, the power of like CRISPR screens, they were able to not be limited in like one gene at a time. Is metabolism kind of limited by one metabolite at a time? Are you kind of relying on just an output, or what are the kind of the future, or their limitations to chemically synthesizing the metabolite that you want to look at, because it happens so quickly? Are you kind of limited in like stability if you wanted to, rather than just knock out a gene and the metabolite not be there? Are there limitations in like the super duper long cholesterol esters?
Yeah, that's really good question here. I think you're referring to full CRISPR screens.
Yeah.
Yeah, so there are different aspects, and I don't think one is better than the other one. Jason we always have these discussions with him, where let me just tell this sort of side story, and I'll come back to the question. Well, we always talk about, he always said, like, genes are sort of more important than metabolites, and then he says, I tell him, well, if you think about it, metabolites make these make genes, and I talk to proteomics people about the same thing, and they say, well, looks like small molecules want to be part of something bigger. They want to just be small molecules alone, right? But then I tell them, like, yeah, but they do that to make more small molecules. So it's always like a back and forth, and none of us is right. It's a systems problem, right? We're doing systems biology. We cannot just look at it just one omic, right? I think with the CRISPR screens, it's very complementary. The CRISPR screens can do, it's fast in terms of it can give you essentially all the genes, but there are many essential genes that you cannot look, right? You can inhibit them and they could be important, but if it's essential, it doesn't work. In metabolism, there's also a lot of redundancy in terms of genes. So, for example, I showed those elongases. There are actually 7. There are only 5 listed. Ceramidases, there are 6. So if you knock one out, there's other 5 that can do the same job. So you never seen CRISPR screen ceramidases, and we know they're really important in cancer. So there's that aspect as well, how it's complementary. When we look at here, every time you do this type of metabolomics experiments, you grow cells. At some point, you stop them, lyse them, and get the metabolite. So it's a snapshot at that particular time. You don't have the temporal aspect of the CRISPR screens. When we do knockouts, that would be more like a follow-up experiment to see, okay, if I knock this out, what happens? So that's more like a follow-up type of experiment, right? But when you get a cell, like we detect all of these molecules all at once in the same not the same run, but from the same sample, right? So I don't know if that answers your question. Give me like a lot of pros, cons, or like what can one technique tell you than the other one. It could be that you see a particular gene in a CRISPR screen and in metabolites, you don't see that metabolite changing because there's rewiring, but it would tell you where the bottleneck or what's happening maybe downstream or upstream that the CRISPR screen wouldn't be able to tell you at the same time. So there are different. There's no like right answer like what would be best. I think they're really complementary at the same time. And the more we work with Jason, the more we realize that it's really like good to have both. And we're working right now and generating what he's been doing with the HAP1 knockouts, doing that on metabolomics. We're going to start doing that. We got 30 knockouts that we're going to profile in the next couple of weeks and start adding that to understand that better. Like what are we missing in terms of what the effect of one gene is on the metabolome. So I don't know if that answered.
Yeah, absolutely. Thanks.
Anthony.
Are there plastic tablets that are hard to ionize in gas phase, or are there different ionization sources that you have to complement?
Yeah, great question. So the approaches we use are different types of chromatographies and extraction methods, and there's not one analysis that we're able to give you all the coverage, right? We use 2-3 type of chromatographies to get around this number of metabolites. And if we add other type of analysis, we would be able to expand that. So in terms of ionization, there are molecules that are really hard to ionize, like squalane. It's just carbons and hydrogen. So there's no oxygens, nitrogen, sulfurs, anything like that would retain a charge which you need for mass spec. And there's sources for that, like APCI, corona discharge. And we just, it's funny you mentioned that because I've not really worked much with them in my from my training and my career. But we just started doing that to measure squalene for a project with Linda Penn in Princess Margaret. So there are other ways that you can get those molecules to ionize and to measure them. I try to, the coverage or like the type of those molecules are like so much smaller. So you would do a big analysis for like less return per se in terms of number of molecules. So we stay more on the ESI to ionize more molecules that could retain easier a charge. But there are ways to look at that, like APCI and corona, chemical ionization unit corona discharge.
So I have a question as well, Rafa. I mean, the concept of the dark metabolome is fascinating. And I was wondering if you can explain, especially for the students, but for me as well, how much of work would go into a campaign of identifying a completely new metabolite that hasn't been seen before? Because you think about it from a protein perspective or a gene perspective, and we know there's certain steps you have to go through, and there's a crystal structure and this and that. So tell us about like how much work is that actually?
Yeah, it's a good question. It will depend a lot on how complex the molecule is. If you think carbohydrates, right, they'll all have the same type of, all the rings have a very, very similar structure. The only thing that changes most of the time is the hydroxy group pointing up or down. So that is really, really hard, right? They're connected as a polymer, but if the hydroxy group up or down, like that's hard. If you fragment that, you will not show much of a difference. So that would be challenging. There are ways that you can try to do it enzymatically and see if with an enzyme specificity try to determine what they are. So in my postdoc, to give you an idea, I identify got really, really close, and I'll tell you what we saw. I was doing an experiment with macrophages, and we were giving it some activating when you're TLR4 with LPS, and giving them some metabolites that are known to be immunoregulators. And we saw a big change in these molecules, and it was the fragmentation pattern can tell you quite a bit of what chemical structures or substructures are there. So you don't start from scratch immediately. You always have an idea of what components could be there. And it looked like a molecule very, very similar to S1P, sphingosine 1-phosphate. I think most of you know that's considered a very important molecule, right? It regulates so many things. I was just modifying the phosphate group. Some of the things that I did to prove that it was real, I used isotopic labels for the sphingosine backbone, like serine, palmitic acid, and then some other component that was attached. So I knew what the different parts were. I wasn't quite sure how it was attached at the end, but I was pretty close. But synthesizing the molecule was hard. I'm not the best synthesis person, but we even got some collaborators at Scripps to help us out. And at some point, it got so complex that I don't know if you know Barry Sharpless, a Nobel chemical, organic chemist Nobel laureate. He started helping us trying to make the molecule, and some of his postdocs couldn't do it. It was really hard to do it. We got close. We have enough information to sort of say this is new. We don't know exactly what this looks like, but if you think about it, one of the best chemists in the world, organic chemists in the world, we couldn't manage to get it. So that's like a sad story because we didn't get to the end, but we got some data, and it was like 6 or 9 months of hard work. In other cases, it can be a little bit simpler. It's just smaller modifications that you can do. I would say the hardest part is probably getting organic chemists to help you or be willing to spend some of the time helping out with that, because the type of papers that they're interested in are not like, oh, you make this molecule with like completely easy to do reactions. So yeah, it's a really broad spectrum of like how easy or how hard it can be, and how close do you need to get to show that it's a new molecule. But yeah, again, many of the molecules at the same time that we think are unknown, like I showed you these long chain, ultra long chain fatty acids with the cholesterol esters, they're not a database. But once you look at the fragmentation pattern, you can identify and you can sort of go to the whiteboard, and it's not that much work. So there's a really broad range of how much time and work goes into that.
It's very exciting because there's a lot of new discovery in what you do, and it's also like being a detective. So it's kind of fun actually to think about. But you're right. It's And one more question to follow up a little bit on Neil's question is about the temporal nature. As you were speaking and answering Neil's question, I was thinking about how I once graded I was an external examiner for a thesis that was all about identifying new protein-protein interactions. And I remember going through all the different techniques that the student applied and looking at the pros and cons of all the different techniques. And there was one thing that almost no technique was good at capturing, and that was extremely transient protein-protein interactions, right? And it's like you mentioned earlier, like capturing that one snapshot in time. And then sort of layering upon that. So what if something happens in such a short time frame that it's hard to reproducibly capture multiple times? And then on top of that, you also mentioned conditions. So I imagine if you grow cells in slightly different media conditions, then they're going to generate different metabolite profiles, right? So to some extent, I just wonder about how do you standardize for a lot of that kind of variability?
So for the Atlas, we're using the exact same batch of FBS. So, and we profile and measure the FBS with the same conditions. So we know what is there. And if you was found FBS, we see then later in the cell lines, we have all that information. Media-wise, we only use, I think, 3 types, and it's very well defined. There's very slight differences, so it's a little bit easier to control. We was doing, excuse me, in parallel the exact same hub once every time we profile something new, because batch variabilities and things like that. So we're doing this as consistent as possible, and in some way, some of the cells that we've gotten from, for example, from you, we want to like then go back and culture those using our 6-well plates, using like very defined the media that we have in place, the same times, scraping the same way. So we're trying to keep this as consistent as possible. But you're right, some cell lines need RPMI, some cells need DMEM, and there's a reason why we researchers use that media for that cell line, right? So we kind of just put all the cells in the same media, not only because they would not grow, but also how useful would it be if I show you a profile for this cell line in RPMI, but nobody grows in RPMI. In RPMI, it would be not very useful. So some of the things that we know is levels will change, but it doesn't seem that completely different molecules would be generated because it's very well controlled, and we give them the exact same FBS.
Right.
So that's how we try to keep it as tight as control as possible.
Are there any other questions? Ali, please.
So I'm not a chemist, I'm not, so immunologist, so this question might be naive. So for metabolite, if we go back to tumor microenvironment, you would right now culturing in vitro, yes? Great. So considering that tumor started years ago, and they have to go through this whole battle with the immune system and all those process, and eventually become solid tumor and establish a small niche of tumor microenvironment. And at that time, the tumor really thrives in an environment of very low oxygen, very like relatively high level of acidity and high level of oxygen capacity, to get the glucose. That's very different than
Cell culture.
them. And my question is really how relevant that would be in a process of the tumor microenvironment, which probably the tumor has to learn eventually to either produce or consume certain type of metabolite to one, sustain growth, and second, to suppress the immune system to some degree. So I'm just wondering that maybe my question is irrelevant at that case, or maybe is there a connection between that in vivo tumor microenvironment and when it comes to metabolite, and in this in vitro, that you have 20% oxygen probably, or 19% oxygen, or whatever, which probably never happened, and probably super saturated glucose, which probably
Sure, very different conditions. So no, that's a really good question. We know that there are going to be differences, right? As soon as you change the conditions, there are going to be differences. What we observed is it matters more whenever you change the conditions. It seems that levels of metabolites change, but not new molecules appear like that, or are just completely unique like this type of biomarkers, or like they would change the fatty acid composition for that lipid class to change so drastically. So we're not focused with this approach. We cannot really, we shouldn't focus on 10% increase of this metabolite here and there. That doesn't really tell us much. We're looking more like what are the big changes here, and I think for that it is more relevant. Now, when you look at the tumors, yeah, of course it's going to be very different, but in the tumor and the microenvironment, you have so many other things going on at the same time, not just this particular cell line, but it's a good place to start. Like what is happening in all these cell lines, and then we can always do follow-up experiments. Looking at the tumors, does this hold or not? Is this something like that we can apply more broadly? But at the moment, we don't even know what is the difference between hepatocyte or hepatocyte cell line versus group 3 medulloblastoma, right? So we don't have the information transcriptomics wise. We use that data all the time. Proteomics, we use all that data a lot, and we know that if you go back in the tumor, it wouldn't, it would not be quite the same. But it's a good place to start to help us understand what metabolites or how metabolism is regulated.
That's why we need to build the atlas in the
We need
first
to
place.
build the atlas. We'll gain more information as we go, and you can always go back and look at the tumors and see what holds and what doesn't, right? But you're absolutely right.
I think Ali is asking you because he wants you to do in vivo metabolomics, since they
Yeah,
already do in
right.
vivo CRISPR. So maybe we can talk about that at dinner.
Great, yeah. Really,
Yeah, I was going to ask a kind of a follow-up to Ali's question, but thinking almost in opposites. When you go from in vivo to in vitro, are you afforded new opportunities to ask biological questions that will be masked in your in vivo system? So I think about stem cells. We can grow a tumor. If you take the tumor out and transplant unpurified tumor cells into another mouse, you probably require 100,000 or 1 million to get a tumor. But in certain culture conditions, we can enrich for tumor initiating cells, for example. So is there an added advantage to your in vitro comparisons where you can modify the media conditions to say select for stemness and see how there are metabolic consequences of that?
sure, you have more control on that experiment, right? I think at the end, it depends what the goal is.
Yeah.
Both have like pros and cons. But yeah, definitely you have more control. You can like go specifically and make those changes there, because even in an in vivo, you might want to treat with a drug, how much is being delivered there, or how much is other, could it be that drug metabolized by the liver or somewhere else? So it definitely, the added control is definitely a plus, right?
And just as a follow-up, do you think that, so the presence of serum, for example, versus non-serum, changing the metabolic profile, do you think that's always just an ingredients thing? Or do you think maybe the presence of growth factors in serum might cause differentiation of the cells? And then through differentiation, they alter their metabolism. And you might think, oh, because they're in serum and the serum has these metabolites, now they're making these metabolites. But when in fact, it's a metabolic result of a changing phenotype.
Sure, there's something to that too. But if you think about it, any cell in the body has serum, right? In vivo, there's no way to do that without serum, and your serum will change a lot throughout the day. After you ate, at night, it's constantly changing. We think keeping using the same batch of FPS, at least we're keeping things consistent, but you cannot just isolate it that way, right?
All right. Well, I think Rafa will be here for Daniel. Why do you want to ask one last question?
Yeah, when you're trying to figure out the structure of that compound in your postdoc, did you apply were you applying a machine learning approach?
No, it's one of the things where when software has improved a lot to get you identifications faster without having to look at the data. I think I was maybe like the last generation where the softwares like really were not that good. Now it's so much better. So now the students in the lab, I have to teach them and sometimes tell them like, don't use it and let's just do it manually so that they get that feeling. At some point you see the spectra, and I always tell them and maybe realize like what the age difference is with the students at the different generations. When you look at the MS2 spectra, you just see peaks and numbers. When you do it for a while, the numbers tell you what it could be because you've seen it, right? So I was telling them, like, if you look at the movie The Matrix, when you sort of see the green code, then you don't need to interpret the green code, but you know what it is. And interestingly, half of my students knew the movie The Matrix and the other half did not, because you realize it came out whenever they were like 2 or 3 years old. But But yeah, you know, you sort of do it manually. This one, it was very clear, like it was this fingered backbone fragments are very characteristic way, so I knew that. And then I knew the phosphate too was like 98. So I was like, okay, wow, this is interesting. What else is there? So I already had good understanding of like what that looked like.
That's fascinating. So machine learning couldn't necessarily replace that intuition.
Well, it's
Yeah.
it's getting better. It's getting better,
I think there was programs.
but we don't
that
have
could-
enough MS2 data
Yeah.
like to train the models. We need to get more of the diversity because the unknown molecules have structures that we are not haven't seen yet. So you cannot train the model on that. So it's
Yeah, I was in another lab that I was working at before, we're using a lot of MS2 peaks to try to like piece back together the compounds through machine learning.
mhm.
So that's what I was
Yeah.
wondering.
Thank you, Daniel. Well, thank you, everybody. Let's thank Rafa one more time. Thank you, everyone, for coming. So, just so we'll